Hello, this is Dr. Jenkins. This is the Senses Lab PowerPoint. The idea here is to just review some of the primary structures that we would be reviewing in lab. It's obviously not the same, but I just want to it helps sometimes to be able to hear someone's voice go through it and talk through some of the most important parts. So here we go. We'll spend most of our time going through the anatomy of the eye for vision and the ear for hearing and equilibrium, with a couple notes about general versus special senses. Here we go. This lab is focusing on special senses, but it's very worthwhile to just make a point that even though our special senses, you know what our special senses are, right? They're limited mostly to our head and they have very specialized functions. We also have generalized senses, and these are in use all the time, all throughout the body. So you think about specialized nerve endings, especially in the skin, muscles, joints. We also have specialized nerve endings in the viscera or our inner organs, for example, lining the stomach or in the kidney. And these are constantly sensing information about certain things, pressure, pain, temperature. So we always have these going on. But this lab is about the special senses specifically. All right, here are our special senses. We're going to be talking about vision and the ear. You'll notice the cute dog pictures. I always feel like at this point in the semester, or towards the end of the semester, some cute dog pictures can cheer us up a little bit. Okay, so here's the eye. On your objective sheet, you'll see the structures you're responsible for. Obviously, many of these pictures have more structures than you need to know. So as always, make sure that you're paying attention to the structures listed on your lab objective sheet. When we talk about the eye, we talk about tunics or layers. So we basically have 3 layers. We have a fibrous layer or outermost layer. We have a vascular or middle layer, and we have an innermost layer called the internal or sometimes sensory tunic. So that's how we're going to discuss the anatomy. This outer white part is the fibrous layer. The vascular layer is the dark maroon layer, kind of in the middle. And the internal layer is this yellow layer. So that's how we're going to frame our discussion. Before we go through the structures in each of the tunics or layers, just want to make a point that we have fluid in the eye, and there's really 2 types of fluid. If we were to look in the anterior chamber, let me use my fancy little pen here. I'll use blue because it's fluid. In the anterior chamber, there's a more liquid or aqueous fluid called aqueous humor. The anterior chamber, as this picture shows us, is really from here to here, but the fluid is mostly in the more anterior part. So in the anterior part of the eye, we have a more aqueous fluid called aqueous humor. On the other hand, in the posterior chamber of the eye, which is really everything here, this yellow, more gelatinous material is called vitreous humor. So in the more posterior regions of the eye, we have a more gelatinous fluid called vitreous humor. And the job of the vitreous humor is gelatinous to help keep the shape of the eyeball. Give shape and structure, keep the shape of the eyeball. Okay, so here we're going to go layer by layer. Let's start with the outermost layer. I'm going to switch to black again. The outermost layer is called the fibrous tunic, and as we said before, this thicker white part is what we consider the fibrous tunic. It is strong. It has to be because we want to protect the eyeball, okay? The large majority of the outer layer is made up of the sclera. So this whole part, all the way around, a brief break for the optic nerve to come out, and then all the way around is the sclera. It's the actual sclera itself is just the outermost part, and it's kind of thin, but it's very rubbery and tough. So in the cow eye dissection, the beauty of that is you get to really feel the sclera. And even though the outer sclera is relatively thin, it is rubbery and tough. The example I use to describe it too, it's almost like the outer covering of a racquetball, rubbery. Now, as we get to the front part of the eye, you can see that if we follow the sclera around, we see a little break here where it changes color. It goes from being more of a blue here to a little bit more green in the front. Same thing here, we see a little color change. So just in this area, in the anterior part, that sclera gets thicker. And the thickened sclera, only in the front, is called the cornea. That's why it says here enlarged. So all the way around is called the sclera, right? All the way around is called the sclera, except for the front. And these brackets are showing you that. It's just a little bit thicker. Because it's thicker, it has a little bit more of a specialized function. It's called the cornea. If you're looking at someone, the white of their eye is the cornea. All right, good stuff. Then we go to the middle layer, or vascular tunic. There we go. The vascular tunic here is shown as being red. Got the black color here. So here, the choroid, or that middle layer, is shown to be red. Shown to be red here. And then red all the way around. Oh, there we go. Choroid. In the cow eye, it tends to be a darker black or darker blue. Here it's shown as darker red. No matter if it's a human or a cow, it's a darker pigmented layer, the choroid. Goes all the way around until we get to the front of the eye, right? Goes all the way around here. As we get towards the front of the eye, in the middle layer of the eye, we have some specialized structures. So let's start with the iris. The iris are these colored extensions that come out. And there's one on both sides, the iris. This is the colored part of someone's eye. The space in the middle is known as the pupil. So you'll see that the iris only goes a certain distance, leaving a space in the middle, and that space or opening is called the pupil. You know this, we can constrict or dilate to make the pupil bigger or smaller, depending on whether we want to let in more or less light. Pupil is the space. Surrounding the, let's see, what else we got? Ciliary body. So what we're going to see in a minute is a very important structure called the lens. And much like the lens of a camera, the lens here focuses the light. So as the light rays come in, the lens can bend its shape to focus or refract the light back upon the retina. But what's holding the lens in place is the ciliary body. And the ciliary body are these muscles. Ciliary body are the muscles. And then, even though it's not listed on the left, you should also pay attention to the suspensory ligaments. So the ciliary body are the muscles. And then in between the actual muscle and the lens are these strings. And those strings are called suspensory ligaments. All right, so we have the lens in the middle. The ciliary body are the muscles attached to the wall of the eye. And then we have suspensory ligaments that connect the lens to the ciliary bodies. A lot going on there, but good stuff. All right, and then the inner lining is called the inner or sensory tunic. And its primary feature is the retina. Here it's shown as being a yellow color. So all the way around that inner lining is called the retina. In the cow eye dissection video, you're going to see in the cow eye, it's almost a beige color, the retina, and it's very sensitive tissue. So that's why it says here, delicate. And the importance of the retina is that all along the retina are the rods and cones. And these rods and cones, we'll talk about next, the rods and cones, those are the specialized cells that are able to process the vision. In the retina, however, there is one point where there are no rods and cones, and that's called the optic disc. The retina is continuous. The retina goes all the way around, takes a little gap so the optic nerve or the blood vessels can leave, but it's continuous. But you'll see something called the blind spot. And in this little region, it's marked here. You see a little hump here. So in that region is called the optic disc. In that region, there are no rods and cones. There's no rods and cones there because that's where the optic nerve's coming in. But it's called the blind spot because what if light rays come in, are refracted or focused by the lens, and what if those light rays happen to get refracted back here? If they get refracted to the blind spot, you're not going to be able to process the visual information, so it's our blind spot. There is a part in our peripheral vision where we do have that blind spot, just because in that one small region there are no rods and cones. I mentioned rods and cones. Let's define them. Here's an actual look at the histology of the eye wall, so we can do it by layer. Here is the outermost layer, fibrous tunic. Here's the middle layer, vascular tunic. And then this whole thing is the innermost layer, which contains the retina. And we have these specialized cells, rods and cones. And they populate a large part of the retina. Some of the differences between the two, rods are more important at processing visual information at low light, for example, when it's nighttime. And cones are more responsible for processing color information. And the way that I've always remembered cones for color is that they both begin with C. All right, moving right along here. Other things in that inner lining that we should be paying attention to. We have the macula lutea. And it is labeled here, macula lutea. So imagine a region. And just like we talked with the optic disc, there was a little oval region. There's a darker pigmented region, sometimes towards the side of the eye. Sometimes it can be a little bit more towards the center. But either way, we'll see a dark pigmented region. And that dark pigmented region is called the macula lutea. Inside the macula lutea, inside that is a smaller area called the fovea centralis. It's in the middle of the macula lutea, hence the word central. So the overall area is called the macula lutea, but the centralized part of that macula lutea is called the fovea centralis, and it has a bunch of tightly packed cones. So it's a very specialized area where we have a lot of cones and we can process a lot of colorful visual information. Maybe you've heard of macular degeneration. That would be degeneration of the cells that make up that dense region called the macula lutea. All right, one last comment. Just as an example of something that could go wrong, we have glaucoma. You've probably had a glaucoma test if you've gone to the eye doctor. Glaucoma is typically a result of high pressure. So we imagine there's a normal amount of pressure inside the eye, but if that pressure gets to be too high, then we're putting excessive stress against the retina, and we're putting excessive stress against all the structures of the eye. And one of the things that can happen is with that high pressure, we can damage the optic nerve. And if we damage the optic nerve, even if we're still getting light rays coming in, and we're still able to process some of them, if we damage the nerve that takes the information to the brain, we're not going to be able to really see because we're not having the connection between the eyeball and the brain. All right, this I put on here as a way for you just to be able to put it all together. So on your own, it would be very helpful to go through the order through which light rays come in. Cornea, pupil, lens, retina, optic nerve. And the optic nerve is what takes the information to the brain. A couple of things to point out. We notice that the information from the left eye crosses over and goes back to the right posterior part of the brain, and the information from the right eye comes back and goes towards the left part of the brain. We also notice that by the time we get to the brain, we're receiving combined information. So actually, some information from the left goes to the right, but some information also goes to the left. So either way, the occipital lobe, if you remember from earlier in this semester, the occipital lobe of the cerebral cortex is the part that processes vision. So that's where the information from the optic nerves is going to go. Each part of the occipital lobe, the visual cortex, is going to receive mixed information from the right and the left eye. I've got a link here. It's also on Blackboard, a link for you to watch someone dissect a cow eye. You should really do it. It's cool. And then I got some pictures here for you to see. The pictures are one thing, but I really recommend going to the video. It's a short video. All right. Let's talk about hearing, another specialized sense. In the case of the ear, we're going to be talking about the anatomy. Instead of layers, outer, middle, inner, in the ear anatomy, we're going to work our way from the outside of the ear in. So the first part we'll talk about is the outer ear. Then as we move further in, we'll talk about the middle ear. And then as last, we'll talk about the very most visceral or inner parts of the ear, called the inner ear. Here we go. The outer cartilage, we know it to be elastic cartilage, because we talked about that in the histology chapter, didn't we? Of course we did. The auricle is the outer ear cartilage, outer ear. Yep. So this is going to help, if you've ever thought about, well, why on earth do we have this cartilage there? It actually helps to funnel sound waves into the ear. And let's see if I go to the next slide here. I do need to add one thing, but that's the beauty of the pen. Let me add it right now. We're going to add external auditory meatus. Meatus means canal-like opening. So the actual opening, the opening through which sound waves come in, external auditory or acoustic. Sometimes you'll see it as it is in the top picture, acoustic. But the name here really tells you what it is. It's facing the external part of the body. It deals with auditory or acoustic sound waves, and it is a meatus, or narrow opening. So really, the external auditory meatus is the ear hole, if we wanted to be really specific and simple, ear hole. As sound waves come in, they're going to be gathered by the auricle. Then the sound waves are going to travel through the external auditory meatus. And then the sound waves are going to enter the auditory canal, or the ear canal. But a better, more appropriate term is the auditory canal. Then we get to the middle ear. Once we get to the middle ear, these sound waves that come in are going to first hit up against the tympanic membrane. In other words, it's our eardrum. But certainly in my class, people wouldn't say eardrum, they would say tympanic membrane. And it's a thin little membrane, much like the top of a timpani drum. It's a thin membrane that vibrates. So as these sound waves come in, it vibrates the tympanic membrane. And then guess what? Those vibrations are going to be transferred to the ossicles, which are the smallest bones in the body, malleus, incus, and stapes, or hammer, anvil, and stirrup. One leads to another. So the vibrations from the tympanic membrane will first vibrate the malleus or hammer. And then as the malleus or hammer vibrates, it'll vibrate the incus or anvil. And lastly, as the incus or anvil vibrates, it'll vibrate the last ossicle called the stapes or stirrup. One other functional anatomy of the middle ear is the eustachian tube or auditory tube. We see the E got cut off there, I'm sorry. And that's this tube right here. So we have a canal, just a canal for air, that connects the middle ear all the way down to the pharynx. And this is it's a tube that is continuous from the middle ear to the pharynx. And of course, the pharynx if we work backwards, goes back towards our mouth. So the function of the auditory tube or eustachian tube is to be able to maintain the proper pressure, to maintain or be able to equalize pressure. Everyone's experienced this. You go up in an elevator to a really high floor and you start to maybe sense that your ears are gaining in pressure. But even before you sense that, you may automatically just open up your mouth as a way to kind of pop. Or sometimes people will chew gum. So the idea is if we go up in elevation, we very well may increase the pressure in our middle ear. And if it increases too much, it could be bad, right? It could damage some of the middle ear structures. So one way of relieving or equalizing that pressure that builds up in the middle ear is to open up your mouth. Because then some of the air that's helping to increase the pressure in the middle ear, some of that air can actually come out the mouth, thereby relieving or equalizing the built-up pressure. Okay, one last structure of the middle ear to talk about is the oval window. And it's the space right here that's in the middle of the stapes. So we had the malleus, incus, and stapes. And the space in between the stapes is called the oval window. It's more than a space, it's actually a membrane, and it's at the junction between the middle and inner ear. It will continue the vibrations from the stapes to the inner ear, which is our first inner ear structure is going to be the vestibule. But importantly, the oval window, not only does it transmit the vibrations, but it significantly magnifies the vibrations. Even up to 10 times, if not more. So it amplifies those vibrations as they're about to enter the inner ear. All right. Let's talk about the inner ear. I think it's easiest to break the inner ear down into three main parts. We have the cochlea, which looks like a conch shell. We have the semicircular canals, which look like, semicircular canals. Semicircular, I'll put an S for that. And then one that you may not be familiar with, at the base of the semicircular canals is the vestibule. So we have the cochlea, the semicircular canals, and at the base of the semicircular canals is the vestibule. Let's talk about each one. The cochlea, not hard to identify it in this view. Cochlea looks like a conch shell. If we were to look more in depth, so if we were to take a cross section, and that's why I like this picture, because it takes a cross section of the cochlea, and if we magnify it, it looks like this. We've got a bunch of chambers, and it has some gelatinous fluid in it. So we have these chambers. There's one chamber. Here's another chamber. But what we're most concerned with is this blue part. And the blue part is called the cochlear duct. It contains fluid called endolymph. But one of the specific structures within the cochlear duct, so imagine the whole blue area is the cochlear duct. So if the whole blue area is the cochlear duct, one more specific structure within the cochlear duct is the spiral organ or organ of Corti. And if you look more closely, I think I have it on the next slide. Yeah. If you look more closely, the spiral organ happens to be this particular row of cells. So within the cochlear duct, or the whole middle blue region, we have a special area of cells called the spiral organ. And this is what's going to do the function of the cochlea. Because the function of the cochlea, as we're going to find out, the cochlea is what's going to help us process sound waves. Just like the retina of the eye helped us to process light rays, it is the cochlea of the inner ear that helps us to process sound waves. And how does it do it? We have these specialized cells that are in the spiral organ, and on top of those cells are little hairs. So on top of each of these little cells are hairs. And as the sound waves come in, Imagine the sound waves coming in from the external ear, middle ear to the internal ear. Those sound waves are going to vibrate and move the hairs. And as the hairs move, the cells beneath will be able to take that movement as information and then send nerve impulses or action potentials to the brain so we can actually process it. Good stuff. Here's an actual look at what these hair cells look like up close. I think it's really cool. So again, as the sound waves come in, the sound waves are going to rustle the hairs and move them. And then the cells underneath will send information to the brain about that movement. How much movement was there and what direction? And that's how we're able, as our brain, to make sense of that sound. Just like you did for the eye, make sure that you can be able to trace sound waves coming into the ear through the auricle, through the external acoustic or auditory meatus, into the auditory canal, which vibrates the tympanic membrane, which vibrates the ossicles, which the vibrations are magnified by the oval window, and then that information goes to the cochlea, and more specifically, it goes to the organ of Corti. And then, of course, that information is going to go to the brain via the vestibulocochlear nerve. The inner ear also plays a role in balance, and I'm sure that you knew that. Let's just talk about the specific structures. The cochlea is the part of the inner ear that process the sound waves. So that leaves us the two remaining parts of the inner ear, the vestibule and the semicircular canals, that help with balance. And the vestibule is more functional in helping us to maintain balance at rest. That's what static means. So the vestibule functions to help us maintain balance at rest, while the semicircular canals help us to maintain balance while we're moving. Let's talk about each. So the vestibule. The vestibule, as we've already said, is located at the base of the semicircular canals. It actually has two parts. So we can see that there's one little circular part here, and there's another circular part here. This circular part is called the saccule, and this larger circular part is called the utricle. But collectively together, they form the vestibule. All right, so we're clear on that. We got two parts. Check out my fancy eraser. All right, let's talk about some more specifics. Inside the utricle, so we're going to be focusing more on the utricle, there's an area of specialized cells. Excuse me. Pardon me. There's an area of specialized cells in the utricle and the saccule, actually. Excuse me. Called macula. So we're going to be talking, actually in both areas, there's specialized cells called the macula. And we're going to see a picture next. But the macula has these important structures called otoliths. And otoliths are really jelly-like stones, kind of calcified pieces. Let's look more detailed. So here we see a magnified view. We see the utricle is outlined here, and the saccule is outlined here, which together they form the vestibule. And in the walls of each are these specialized areas of cells. They're shown as purple. The macula. The macula has these otoliths, and they really do, up close, kind of look like jelly-like stones. So how does this help us to maintain static equilibrium? Well, if you change position, if you were to be hung upside down and you were no longer moving, you were hanging upside down but you were not moving, and you closed your eyes, so you didn't have any visual cues, wouldn't you still know that you were upside down? Of course you were. And that's a product of the otoliths in the macula. So as you move, the otoliths move. Excuse me. If you take a jar and put some stones in it and you turn the jar upside down, don't the stones move? Yes. So as you move, the otoliths move. And these hair cells underneath sense the otoliths moving. And then these cells can send information to the brain about that movement. And that's how you know that you are at an abnormal position, and hopefully you can correct yourself. All right, really cool stuff. Lastly, we have the semicircular canals, and they form 3 semicircular canals, 3 of them. What do you notice? There's one in each plane. There's one located, if you want to practice along with me, this one is located in the plane that divides us into right and left halves, sagittal plane. This one is located in the plane that divides us into front and back halves, frontal or coronal plane. And this one is located in the plane that divides us into top and bottom halves, or the transverse plane. Excellent. So what that means is, because they're in each plane, no matter what direction you're moving, front and back, side to side, twisting, there's a semicircular canal in each plane. So no matter how you're moving, there's a semicircular canal that's going to be able to pick up that movement. So semicircular canals help us to maintain balance or equilibrium while we're moving. That's why these pictures are showing you a gymnast. If we look at the structure, within these semicircular canals, there are elevations in the walls called crista ampullaris. So there's elevations inside the wall, crista ampullaris. And of course, we have some fluid called endolymph. Check it out. We've got some fluid flowing around. As you move, guess what? The fluid moves. As you move, the fluid moves. And as the fluid moves, it moves these hair cells, which of course means that the cells underneath can send nerve impulses to the brain about the movement. So we notice a trend here. We're utilizing hair cells in either fluid or otoliths. Here's another look at it. Just saying the same thing, but putting it down in words for you. There's a lab activity. So here's a video for you if you want to look at an otoscope. They attach an otoscope. They attach a camera to an otoscope, and you get to see what it looks like going through the auditory canal. It's pretty cool. And then the rest is up to you to study. Okay, good luck, everyone.